As we've heard, hepatitis B and hepatitis C are global health problems, and we are interested in trying to make animal models to study viral hepatitis in vivo. And so this is my cartoon, my favorite cartoon, which is essentially trying to say, how can we make animal models resemble humans, and how close can we get to that idea? So the animal models that have been described in the literature have been essential to establish the drugs that are being used in the clinics today. And these include transgenic animals as well as AAV, HBV transduced mice. These animals are very important to look at viral replication, but you cannot really look at an immune response, or definitely not a human immune response, since these are mouse-derived immune responses, and it's not a complete viral life cycle that you're looking at. You don't have the cccDNA form. The use of humanized liver mice is interesting because you're using human hepatocytes and you're looking at a full viral life cycle. However, you don't have an immune system in these animals since they are immunosuppressed to be able to accept the human grafts. And the chimpanzee model, as everybody in the room knows probably, was essential to develop vaccine strategies, yet the ethical considerations have limited their use, and their numbers were difficult to be able to have large cohorts. So the differences between humans and mice are notable. One of them is the immune response. The immune cells are very different already if you're looking at the blood. In humans, you have a majority of granulocytes and monocytes that are present, whereas in mice, it's a majority of B cells and T cells. The NK cells profiles are very different. The ligands are different. The inhibitory cure receptors are different. There are also differences in the innate response, the TLR expression patterns that have been interesting for immunostimulatory or regulatory mechanisms, and the FC receptors are different. In the case of the liver, there are also important differences in hepatocyte cytochrome P450 activities, and this is important in terms of pharmacokinetics, the ADME studies that need to be done before you get drugs to the clinics. And for this, you need to study human hepatocytes. And finally, as you know, the virus has specific species tropisms, and this limits viral entry, as well as the replication of the virus and the production of viral particles. So for all of these reasons, we've been interested to establish humanized mouse models. So this is essentially the three models that we develop in the lab. The first one is a mouse in which so all of these animals, are immunodeficient. They're RAG2, IL-2 receptor gamma C knockouts, meaning that they have no T, no B, and no NK lymphocytes. So in these animals, you can graft cells that are human origin. And the first model is what we call the HIS model for humanized immune system. We take the animals at birth and we inject them with human hematopoietic stem cells. And after about 3 months, we get reconstitution of the immune system in the animals. The second model animal is one that you graft with human hepatocytes that we've heard of already, and this is what we call the HUHEP model. And then the third is a combination of these two, in which we inject first human hematopoietic stem cells and then go back to the same animals and inject human hepatocytes, and we call it the HIS-HUHEP model. So to generate the humanized immune system model, we take the newborn animals, we irradiate them, and we inject them with human hematopoietic stem cells. And then after about 10 to 12 weeks, we can analyze in the blood by the FACS of the reconstitution. So here you're seeing human CD45s compared to mouse. You see CD3 and CD19 cells, the CD4 and CD8 populations, as well as monocytes, which aren't shown here. And then if you look in the different organs, you see that you have primary, secondary lymphoid organs that are reconstituted. So we have a full population of hematopoietic cells in the animals. And these animal models have been extensively used by others to infect as models for HIV and EBV, as well as CMV, Ebola, dengue, and a whole sorts of experiments that are listed here. There are a few publications, but it's a full field. In case of the HUHEP models, there are three models that were used historically to study a human hepatocyte engraftment. You first need to create liver damage, and to do this, you can either use the albumin UPA transgene that Ulrika has presented already, which was developed by Maura Dandri's group, and these animals have very high levels of liver engraftment. Over 50% of the liver is chimeric in human, and this is measured with human albumin in the plasma of the animals, and we're above 1 milligram per ml. If you use an animal model developed by Marcus Grumpy's group, which is called the FH knockout, you also get very high levels of liver engraftment. You need to do a few tricks before, which are adding a UPA adenovirus to help the hepatocytes get into the liver and engraft. But the advantage is that you can inject the adult mice because you can cycle the animals on and off NTBC to create the liver damage. And then there's the TK model, which also accepts human adult hepatocytes, and this is given with the ganciclovir treatment, and you also get high levels of liver engraftment. So these humanized mouse models have been used to investigate hepatitis viruses of several types. So in case of HBV, we've already heard some of the studies that have been described by Maury Dandry's group and others. In the case of HCV, these animal models have been able to test different antivirals such as interferon, telaprevir, ribavirin, and more recently, they've been able to determine whether receptor-blocking antibodies can be efficient, such as CD81, the Claudin-1, and the SRB1 group. And then, interestingly, I think these animal models can also be used to look at hepatitis E virus, which we haven't talked about that much. This virus is difficult to model because in these animals, normally it's an enteric infection, and yet in these animals, you can't just give it by oral gavage, you have to give it by an IV injection or an intrasplenic injection, and we don't really understand why that is. But the animals can be used to look at the antiviral innate response and viral particle formation. And we'll hear probably a little bit more about these animals that can also be used to test HDV, HBV co-infection, which is a very important health problem. About 10% of patients that are infected with HBV are co-infected HDV, and these animals allow you to study how the co-infection accelerates liver diseases, how it modulates the antiviral response, and to test entry inhibitors. So we have been interested in trying to model not only the human liver, but also the immune response against the virus in the liver specifically. And we were interested in doing this with livers that are highly chimeric, because we believe that you need hepatocytes in large quantities to be able to get a good replication of the viral life cycle. So we inject these BALB/C RAG gamma SERP alpha UPA transgenic animals. First, at birth with these human fetal liver CD34 positive cells, and then we go back after about 8 weeks with adult hepatocytes, and these are HLA mismatched. And then at about 10 to 12 weeks, we're analyzing the blood, the immune system with the FACS with CD45s, as I showed you, and the human albumin to look at liver chimerism. Now, if you inject the adult hepatocytes at a later time point, you don't get co-engraftment. So this is probably due to a tolerogenic liver environment. So if you compare animals that are grafted just with hepatocytes with animals grafted with immune system plus hepatocytes, you get similar liver chimerism and similar kinetics over time. You get 20 to 50% of the human hepatocytes that are engrafted, and so the liver is highly chimeric. If you look at the immune system of these animals, you get similar reconstitution in the primary and secondary lymphoid organs. So here we're comparing animals humanized only for the immune system, called HIS, with the double humanized HIS-HUHEP. And when you look at the liver lymphocytes, it's similar populations. And if you look in the liver of the animals, you don't see any graft versus graft reaction. And this is probably due to the fact that you're tolerizing the liver by injecting the hepatocytes while the T cells are being educated in the thymus of the animals. So several studies have discussed the outcome of chronic HBV infections depending on the viral load. And this first study by Francesco Cesari's group showed that depending on the viral inoculum, you get different outputs and different viral controls. And this important REVEAL study that we've heard about repeatedly also shows that depending on the level of viremia at a very early stage, you get progression into hepatocellular carcinomas that can be extremely different. So we asked the question of how the viral load impacts on the immune response. And to answer this, we infected the animals with different viral loads. So here we're looking at animals with only humanized livers, and the virus is indicated with the line, and the human albumin chimerism is indicated with the bars. In case without any immune system, you get continuous virus replication in the liver, whereas if you infect animals with a moderate dose of virus, you get first in a high viral replication, and then it plateaus out. And in mice that you inject with high viral load, also you get high induction of viremia and then a plateau. If you look in the liver of the animals, to determine whether you have any hepatotoxicity, we can see that the human hepatocyte function is decreased in the animals that are infected with either a low viral load or a high viral load. And this is due to direct toxicity in the liver, and this is revealed with an M65 ELISA that's showing you cytokeratin 18, the fragmented form, which is human-specific. And the viral loads that we're measuring are correlating to the HBs antigen loads and the HBe antigen loads, and importantly, we detect cccDNA in the liver of the animals to levels that are comparable to the chronic hepatitis B patients. If you inject animals with a low viral load, you get 0.1 cccDNA copies per cell. At a high viral load, it's about 2. And the viral replication is the same whether you're at a low viral load or a high viral load. When we look over time, we see that the animals with only liver humanized get increased viremia, whereas in the mice with livers and immune system, you see that the albumin does not correlate at all with the viremia. And when you look at this over time, you realize that there's a control of viral progression in the HIS-HUHEP animals. So if you look in the liver of the animals, you see here in blue the human albumin, in green the HBC core, and in red either CD45, so you have very high levels of liver inflammation that's mediated here with a staining of CD3 in red, but it's mediated by T cells that are encircling the infected hepatocytes, and it's also mediated by the Kupffer cells, the CD68 positive cells. If you look in the animals at sacrifice, so here we're at 3 to 4 months post-infection, the NK cells are increased in numbers according to the viral load that you're injecting, and they're very activated, and they're expressing higher levels of CD56, CD16. And the T cells are also increasing in cell numbers, both CD4s and CD8s in the liver specifically, and they're switching phenotypes from a naive CD45RA to a memory CD45RO phenotype. If you look at their profile of PD-1 expression, we see that only in the liver of the animals, the CD4 and CD8 T cells have a higher level of PD-1 expression compared to the controls, but only in the animals infected at a high viral load, not in the animals infected at low viral load. And we see that this level of PD-1 expression correlates to the viral load only in the animals, again, infected at a low viral load, suggesting that when you infect animals at a high viral load, you're swamping the immune system, and it is rendering it more difficult to get a response that's specific. We wanted to determine what the ligand expression pattern was on these animals, so we looked in the liver at the PDL1 expression, and we see that PDL1 expressing cells are quite numerous. They're both antigen-presenting cells and hepatocytes that can be co-infected, that are infected with HBV. You see here with the green, blue, and red co-stainings. And in terms of the antibody responses, the humoral cells, the humoral immune response, we see an increase in total IgG, and we see antigen-specific antibodies, so IgGs, both HBC core and HBs antibody positives mice. And when you look in the plasma of the animals, we're looking, in this case, we're looking for biomarkers. We see that there is a strong pro-inflammatory response and also some interferon response that's mediated probably by the interferon alpha. We see it with the MIG and IP10 upregulation, but we also see immunosuppression with higher levels of IL-10. And these biomarkers correlate to a certain extent with the viral load. So if you infect animals at a low dose, you see correlation with CCL3 and with IL-10, but you don't see this correlation when you infect animals with a high dose. Yet these biomarkers can correlate with the expression of PD-1 in the CD4, CD45RO liver-specific cells. So this is true for interferon alpha, for IP10, and for IL-10, indicating that the immune response in the liver can be tracked with these biomarkers in the plasma. We were interested to see whether there was any trace of modification of hepatocytes in terms of their differentiation, because there were studies from Jessica Zuckman's group that described that liver progenitor cells, or oval cells, that are present in HBV-positive chronic patients have more of a hepatocellular carcinoma type phenotype. And so we looked for these oval cells, or these EpCAM-positive, CK7-positive cells, and we indeed found in animals that were infected at a very high viral dose that these cells were positive and present in the liver of these animals, and in more numerous than in the animals that were infected at low dose. We tested therapeutic strategies in these animals. We first started with something that is well known, such as Entecavir, to determine whether lowering the viral load could modify the immune response in the liver. And we saw that, and we gave them animals Entecavir, we saw a 3-log reduction in viral load. And this 3-log reduction in viral load with no change in the HBs antigen is indeed sufficient to restore naive phenotypes. So we see diminished numbers of monocytes, diminished numbers of NK cells. The phenotype of the NK cells returns to a naive phenotype in the liver. We see diminished numbers of CD4 and CD8 T cells, and even the PD-1 expression is downregulated in the T cells in the liver specifically. So, and furthermore, when we're thinking of immunoregulation, we see that the number of T regulatory FOXP3 positive cells is also diminished in the liver of these animals. So when we're thinking about these different humanized mouse models, there are many different models that have been described, and they each have their own specificity, their pros and their cons. I think that one of so Lishan Hasu has presented his animal model, and Charlie Rice's group had an interesting model that was also recently published. I think that what we're lacking mostly are antigen-specific T-cell responses that are something that you can correlate to what people are seeing in the clinics. And to address this issue, we've crossed into the UP animal model HLA-A2 and DR2 transgenes in order to get more efficient T-cell education, presentation of antigens and T-cell responses. So to do this, we just repeated, once we had crossed in the HLA-A2-DR2 transgenes, we repeated the experiment, and I just have some analysis of the animals at the blood stage. We see that we have slightly better CD45 reconstitutions. It's quite equivalent in terms of CD3 and CD19 ratios. However, we see a better proportion of CD8 T cells being developed, and this has also been described in animals that are just humanized for the immune system without the liver. And we see that the engraftment of the liver is stable except for this one animal. And so I hope that soon I'll be able to give you the results with the HBV infection to see whether or not we detect these antigen-specific T cell responses. So humanized mouse models can be used for many different things. There are 3 main components that need to be juggled and thought about. The first one is the immune system, the CD45 cells that you're going to develop. We need to improve the myeloid cell compartments. We need to improve the HLA compartment, the HLA presentation, and we need to improve the development of plasma cells. And this is probably going to go through the fact that we don't have any lymph nodes that are well reconstituted in these animals, and this problem needs to be addressed. One way of doing this is by adding back human cytokines and growth factors that don't cross-react between mouse and humans, and that could be lacking in the system. When you think about the liver, we're only addressing the hepatocytes. We don't see any human fibrosis in our model, and this is probably because we don't have any stellate cells, and we don't have any mesenchymal cells that are producing these stellate cells. In terms of the different viruses, you can infect these animals with different doses, and you can also use viruses that are from either plasma or from patients, or from cell culture. But you can also look at co-infections. So you can look at HBV and HDV, or you can look at maybe HIV and HBV, and this is something that we're particularly interested in. Or, as Jake showed us very nicely, you can do Hep B, Hep C co-infections. So these are different future ideas to optimize the humanized mouse models. I think we've gone through some of them. For the lymph node development, we're using TSLP transgenic animals. To increase the dendritic cell niche, we've knocked out the FLIC2 and adding back FLT3 ligand, which is human-specific. human mouse specific, but we knocked out the mouse component, so we only have development of human cells. And we crossed in the human HLA transgenes to improve T-cell development, and we still need to work on the human cytokine cross-reactivity with mouse cells. So I think we'll pass through the summary, and I just wanted to thank everybody in the lab that contributed to this work, and thank the funding agencies, and we'll be happy to take questions at the panel.